Welcome to Honors Spotlight, the podcast where we showcase the incredible opportunities and inspiring stories from the MTSU Honors College. I'm your host, Robin Lee, and today we're exploring the fascinating world of microbiome research. This summer, two of our honors students, Tatyana Martinez and Ian Wilson, took part in an amazing research experience for undergraduates, or REU, right here at MTSU in the Microbiome Ecology Lab. From hands-on experiments to tackling real-world questions about snake fungal disease, these students have an incredible story to share about science, discovery, and the power of research. So let's get started. Can each of you kind of introduce yourselves and share your major, what year you're in, and just a little bit about your academic interest?
Sure, yeah. So I'm Ian Wilson. I'm a senior in biology, concentration, organismal biology, ecology. I'm in my last semester currently. So I am very big on ecology and specifically disease ecology, which is a little bit informative of why I'm in this area of research. I've always been passionate about better understanding the interconnected relationships of organisms. And so I feel like I've gotten to find a really good place here at MTSU to kind of learn more.
Any hobbies outside of ecology?
Well, I like to go hiking, which I don't know if that can really be said as independent of ecology. I enjoy biking. I sometimes bike to campus from my apartment when it's not too hot, which it's been great recently with the weather. Yeah, I don't know. I like really terrible films.
And my name is Tatyana. I'm a third-year biochemistry student doing my honors thesis in the Walker Lab currently. And I don't know, I guess I'd say my hobbies include playing video games and, I don't know, reading a lot of philosophy books that kind of get me, scared for the world. But not, I don't know, that I can make a difference. So, yeah.
Do you have a favorite video game?
Okay, this is really cringe, but Rocket League is I'm obsessed with Rocket League.
That makes sense for what I know about you, yeah.
Yeah, it's like soccer, but with cars.
So how did you each first hear about the research experience here?
Well, I think our introduction to it was pretty similar. We both started in Dr. Walker's undergraduate microbiome scientist program back in spring of, I guess it was 2024. And so we went through that whole spring semester working in that research experience, during the semester and balancing it between classes. And then by the end of that semester, Dr. Walker sort of approached us and saw that we were really passionate about our work and we put in a lot of time outside of just the prescribed hours. And we were really kind of invested, and he kind of proposed that we stay on for the summer to work on this REU.
That's awesome.
Yeah, we saw flyers around first about the REU, and we're kind of interested, and we didn't really know a whole lot about it until Dr. Walker finally approached us about it. And yeah.
Thanks for sharing a little bit about you and what drew you to your academic paths. So what is the focus of your current research in the Microbiome Ecology Lab?
It's a mouthful.
It is.
You should see the email extension. It's really wordy.
We're just going to say the lab from now on. What is the focus of your current research in the lab?
Yeah, so we focus a lot on disease ecology within the microbiome. So looking at who all is there and what this can mean for the disease state of the microbiome and potentially what this could mean for future conservation work. Particularly over the summer, we were really focused on the chemistry and sort of the mechanisms behind it, what is driving snake fungal disease to be so deadly and be so detrimental, and how does this structure the microbiome?
Yeah, a lot of it is specifically with our involvement focused on snake fungal disease, a fungus, and how it interacts with the bacterial and fungal members of the microbiome. So a lot of bacterial fungal and then fungal interactions, and then how that impacts the community as a whole.
Fungal interactions. For somebody who doesn't know a lot about snake fungal disease, how would you explain why this research matters?
The best comparison is the unfortunate times when you get ringworm or something, or you have an actual skin fungal illness. I mean, they're very similar. They're both dermatophytes. They impact us in a similar way, but imagine that a lot worse. The lesions get really intense for snakes, and it presents a really big challenge for conservation of reptiles, which are already vulnerable to our impacts on their natural environment, their access to stable, consistent places to live. And I mean, beyond that, it's a serious matter. Conservation is a big deal because if you take one group of organisms out of the equation in our overall ecosystem, the whole thing can collapse. Like, we're a lot more interconnected than I think people realize. Like, you hear, oh yeah, we're all this like big web of organisms, but it can be really serious. And
yeah, heavy on the emerging like fungal diseases. It's snake fungal disease has been found in like snake populations all across the globe, and it's of a particular concern out here in the southeastern United States, and that's why MTSU is able to be such a good place for the research of this disease.
And just out of curiosity, are there any snakes that don't acquire these types of diseases? Because there are a lot of snakes in the little river by my house, and I don't like going kayaking there because of it.
There is definitely something to be said that certain snake groups are like potentially more vulnerable to like skin illnesses and
we did a research project actually on this question.
Yeah, we were kind of looking at this like lipid composition of different snake species and their snake skin and extracting those and seeing how the fungus grows on like a media that is resembling those lipid profiles. But I mean, as far as I know, it affects,
it's pretty consistent across the board. I think we tested Nerodia, and Nerodia had like the least amount of fungal growth. There's something to be said. And it's just the lipid profile with keratin, which is the main carbon source in the skin. So you can't really adequately capture like the immune response that the snake might have that might better be able to defend itself. So we're not able to entirely answer that question, but we do have some preliminary data to suggest that maybe Nerodia, which is the water snakes, they're a little less resistant or more resistant to the disease.
Yeah. And a lot of it is like different factors that it's hard to kind of control for like vulnerability. Like some people, there's like human skin microbiome is not as strong, and so they have more issues with inflammatory diseases or like, fungal infections or whatever. And so it's the same for snakes. Some groups are more vulnerable than others, but it's not a rule as much as just like differences in populations.
Yeah, and that's exactly what I was wondering about, because I know some people have really bad reactions to certain allergens and other people it doesn't affect them at all, and I don't know how different snakes are from that.
Certainly, I can empathize with that.
What does a typical day in the lab look like? Is there a typical day?
I don't know. The day, it changes so much, because nothing, you can have a plan set in place. Whether that plan will come true is a different question, because so many things can happen, whether you find results that are interesting and you want to run a certain test, or you find something and you're like, that doesn't sound right, like, let's take a pause. Obviously, there are some things that are routine, like especially with a time series experiment like the one that we were doing this summer. There are times when you need to get things done, but whether the rest of your day, like what the rest of your day shapes out to be, is a different question. I know this semester we're going to do a lot of molecular work, which is DNA extraction and sequencing to see like who all is there in the microbiome to kind of get those results for our summer work.
Yeah, I would say like working in the lab is one of the most varied and like kind of exciting work experiences, because you don't get stuck doing the same thing to an extent, like every single day. Like there's always something different you could be doing, or like some days you're writing or looking at the data and coding in our studio or something, but then some days you're working in the hood like from 9 a.m. to 4 p.m. just like moving cultures or like inoculating stuff. And so there's a lot of variety and like a lot of different experiences and opportunities to train on different stuff like molecular work, like Tatyana was saying.
When I interviewed you guys for the MTSU News article about your REU, you both mentioned collaborating with other labs. What is that like?
It's been amazing to collaborate with so many different labs. I think what's interesting is that you're, as undergraduates, or I guess really anyone who's collaborating with another lab, you have to take into account that not everyone's going to fully understand what your experiment is, like all of the details. Be able to explain what your project is and explain your questions, especially to someone who maybe it's not at all related to what they're doing. As a microbiologist, talking to a biochemist like Dr. Miller or Dr. Weissmiller, and being able to have a conversation, because like sterility is a big thing for microbiology, whereas maybe in a biochemistry lab, that's not really something you really have to consider.
You go upstairs to the third floor of the science building and it's like a whole different world. The labs just operate so differently. It's really fun. No, and I want to add to that, like, it's also really nice to get to interact with these other people with different specialties, because it almost feels like you get to kind of like merge their brains into your brain a little, and you get to just like pick them for information. We obviously have most of our experiences in microbiological work, like ecological like work, like that's our background as we've spent like these 2 years in the lab. Just talking to other people with more maybe chemistry slanted expertise, you kind of get to say, well, hey, I have this preconception because I've taken a class about this, and so I generally know what we're doing, but just to be sure, because you know better than I do, and you get to kind of like increase your knowledge on the subject, you get to kind of see their input, and I feel like that's really kind of like, it's really nice to talk to someone who knows a lot about a thing that you don't know a lot about. I don't know, you get to like achieve more than you would have by just like working independently. It's, I don't know, it's very literal, but very enjoyable.
Yeah, absolutely. You learn more.
Yeah.
Isn't the third floor where they have all the stuffed animals on display?
They have them all over the
They're all over.
Yeah.
Some of them are really bad.
They're funny. I remember my first semester walking around the science building for the first time. I kept getting lost, because I feel like that's like a very freshman experience, is like getting lost in the science building. But then I would just like turn the corner and I'd see like the stuffed monkey on the first floor, and I'm like, oh, or I think it's
Where I am.
yeah. I was like, oh, hi, hey, how's it going?
I think the first one I saw, I think it's a mountain lion.
Oh,
I think that was the first one I saw, and I'm like, oh, okay, that's here.
They've had some new stuff too. I was like there's more like birds or bugs or something, or is it like sea stuff? I don't know. It's fun. I'm a big fan.
I need like a giant shark, like in a cage. Would be awesome. So, are there any surprising or aha moments during your research?
I think the biggest thing about like the aha moments is like you in the moment, you're not sure if it's a good aha or if it's a bad aha. And so we had a carbon negative control, or it was supposed to be carbon negative control.
Just to like a media that like has like nothing supposed to grow in it. It's just like basic salts and stuff.
Yes.
Thank you for explaining that for our listeners.
And so we noticed that our bacteria seemed to be growing really well in what was supposed to be the absence of carbon, which everything needs carbon in order to survive. So we expected it to just die off, but it just proliferated. Like it did super well. And we were like, is this a good, like what's happening? So that was when we collaborated with Dr. Miller to really investigate that, and did an SES page, and we found that there was some residual carbon from when we did dialysis, which was just to separate certain parts of our media. I guess what was interesting about it is that in the presence of a lot of this carbon or keratin, like these communities didn't do as well, at least in the preliminary results. But then in the presence of like a little bit of carbon, they're doing so much better. We weren't expecting to see that result because we weren't expecting there to be any carbon at all. But the fact that there was allowed us to glean some more information about maybe that there's an impact on how much carbon is there.
It's really like an experience of like there's like a ton of like little aha moments, like little like once you're like, oh, that's interesting, I wasn't expecting that. And like the rest of the job from there on is like basically just trying to interpret it, because they never really like clear cut like this is like a good conclusion, this is a bad conclusion, because they're all just conclusions. You just sort of have to like make sense of it, double check your work essentially, and just kind of like go forward and say, okay, if this is what we're seeing, if this is what's true, then what does this mean in this context? What does this mean?
It's always a lot of if, then, why.
Yeah.
Typical scientific way to put it. Yeah. That's really fascinating. So it sounds like you've been deeply involved in some meaningful work. Beyond the science, though, this experience was part of a larger program, the REU. So let's talk about what led you to apply and what the overall experience has been like for you.
Well, I know from my experience, I was applying to a bunch of different other REUs to like for my first one, not this past summer, but the summer before. And because I was really interested in getting some research experience, and I was interested in research, I wanted to pursue this further. So I was like, okay, this is like the next logical step. And then when I got the offer from Dr. Walker, after having talked to him about his REU that he was doing and asking about details and stuff, I think I asked him like the beginning of the semester, I was like, hey, so I saw you're doing an REU, like, what's about all that? From there, I kind of was like, through the semester, getting more involved in the lab, like getting more exposed to the research. I had seen other people in the lab and what they were working on, and I was like, oh, this is something I'm very interested in. Like, it kind of feels like the perfect niche. I think like when I interviewed for the undergraduate microbiome program, I'm really interested in ecology and like the way the organisms interact, but like taking it to this like scale of like a microbiome where, you know, you don't really see your microbiome, it just sort of exists on you, but there's this like whole ecosystem just living on you that is a part of your overall self that I found really fascinated, the way that like things on such a small scale interact and have such big implications on the macro ecological scale. I already am exposed to this research. I kind of have like my foot in the door, and this seems like a really great way to like give me more experience, take it to the next level, and, you know, be in something I'm very specifically interested in.
I think it being at MTSU was like a big draw. You can be here before the REU. You get to know Dr. Walker as well. And you've kind of already had some exposure to the lab and the project. And so you can really hit the ground running over the summer. And then you can even carry that on past the summer and into the semester, like what Ian and I are doing with our honors thesis project.
Yeah, because we started that project over the summer, and I got to adapt it into my honors thesis, and that was really helpful to get a lot of work done, like talking with my committee and stuff.
So how has this REU helped shape maybe your future goals and career plans?
I think for me, it's really shaped that I really have a passion for the microbiome and understanding how it plays into the host's ability to defend itself and just like basic immune response. I think it's really, really interesting to see, especially from a medical context. Like, you can take these systems and these experiments that we're doing in snakes and for conservation work, and you can apply them to almost any living thing. You can apply them to humans and learning about things like ringworm, things like athlete's foot, and even beyond that, to understand how the microbiome can affect your ability to defend yourself. And it's always there. So
yeah, I think like getting to do this REU like really kind of solidified that I was interested in research. It wasn't just something I was like, oh, that sounds really cool, like I'd like to get into that, because that was kind of me my whole first two or three semesters at MTSU. I was like, ooh, I really like research, wish I could get involved. I don't know how to do that, because I was just so like overwhelmed with everything, transitioning into, you know, college and being kind of socially awkward. I was just like, I don't really know how to do this. But like getting to kind of like immerse myself fully and like have like concrete proof that, you know, research is something I enjoy, it's something I can do, it's something I'm passionate about, it scratches that itch, was, you know, really satisfying. It feels weird saying itch now that we've been talking about like infections, but yeah, it's really satisfying. And it kind of showed that like now in my last semester at MTSU, I'm like looking at graduate programs to apply to. I'm still figuring that out, because it's maybe one of the most stressful things to comprehend is like, oh, where am I going to go? What am I going to do?
what's next?
Yeah, what's next? Yeah. But it definitely has provided this sense of like, okay, well, I know what direction I'm throwing my stone in. You know, like I know where I'm going to end up generally. So it's, you know, just working out the details now.
So what kind of mentorship or support did you guys receive as part of this REU? And what did you learn about yourself as a student or even as a researcher during this experience?
Over like the course of the two REUs, as far as like experience, I think Dr. Walker has been a really great mentor. He very much like knew where we were at. Like, he had seen us during the semester. He knew that we were really passionate, but we didn't have a ton of like specific experience in what we were about to do. So he kind of like led us into the water, but then like kind of knew the right point, I feel like, to kind of say, okay, so we spent like a couple of weeks now like getting in the swing of things. You guys can start doing stuff more independently, and we'll see where that goes. The more times that you kind of get that experience and then work it out yourself, the better equipped you are to start handling that stuff by yourself and not be so like nervous. I know specifically for myself, I had a lot of those moments where I was just like...
I know he said you guys were basically working at the graduate level because you were doing your own experiments and handling things before, so that definitely speaks to you guys' passion for this project.
Yeah, I think being in the lab for, I guess, how we've been trained in the lab, starting off with like just basic, not even having a question, but just really getting familiar with the lab, and then sort of building that challenge into, okay, now you have a question, let's design the experiment. And then even going into, okay, now we're going to figure out how you can analyze the data and so on and so forth. But he's been very amazing at giving you the right amount of independence without making you feel like you're on your own or you can't ask any questions.
So it's great to hear how this experience has shaped your academic and professional goals. But let's be real, research isn't always smooth sailing. So what challenges have you faced and how did you navigate them?
Well, this past summer experience was definitely a display of not that the project went wrong, but it was just a lot of oh, we didn't expect that. Oh, we didn't expect that. Kind of working out the kinks in this experimental design that we kind of talked over with Dr. Walker. And we were at an experience level, like we were saying earlier, where it was kind of all right, you guys are the first line of defense to kind of work this stuff out and talk back to me if you need any kind of spitballing or whatever, what to do. But this is kind of your project now. So it was a lot of taking ownership of what was going on and kind of thinking for ourselves all right, well, we were trying to do dialysis in this way and this might not work out, so we're gonna have to do it this way. And
it's a very interdisciplinary project, and so it was a lot of stuff that maybe we weren't as familiar with, and so it really brought up that challenge level of reaching out ourselves to collaborators and trying to solve things, and then relaying that information back to Dr. Walker and being okay, so we talked to XYZ, this is what we heard back, and this is what maybe we're thinking as far as a plan for next steps, and him providing feedback on, okay, yeah, that sounds good, or maybe let's try this as well. And a lot of mini experiments and side experiments to figure out, okay, is this really what we're seeing, or how do we interpret what we're seeing?
I think there was a 3-day period where we were just figuring out the best way to evaporate water. So silly. Out of context, but in the moment it's all right, laser focus, we're putting together different parts of a vacuum tube hose with a
little bit of engineering there.
A little bit of engineering, yeah. We're designing all sorts of little prototypes. And
did you have to 3D print anything in Makerspace?
That is a good idea. That would be great. We could rapid turn around that. The number of times we have
print dialysis too big.
Yeah, exactly. Just the number of times where we're oh, I wish we could have something that looked like this and worked like that, or clipped it to this thing, or and we it'd be great if we could just start making our own labs.
Make your auger plugs.
Yeah.
So for students who are listening and thinking, I don't know if I could do something like this, or even for those students who don't realize that REUs are even an option, what would you say to them?
I would say, as someone who still is has to remind myself every day that I am capable of doing it, you are so capable of doing it. It's really I feel 9 times out of 10, your own internal dialogue about it is more critical than anyone you can encounter that will say anything to you. And the people who are doing what you want to do, or doing research, it's not that they are just somehow magically more equipped to handle the type of work that they're doing. It's that they're willing to just do it. If you can get experience, no matter what experience that is, that's helpful. Just building up your base of confidence so that the stuff that you're sweating over now, you look back on it a month or two or a year later and you're oh, that was nothing. I don't know why I was so worried.
I feel you can learn so much even without having traditional training. When I first started in the Microbiome Lab, I didn't have a lot of the coursework to support what people might think you need in order to be in a microbiome. I hadn't taken microbiology yet, but I learned so much about the system, and I learned so much about how to approach questions and generally just lab practices that helped me in my coursework, but also just learning how to problem solve without, traditional training, I guess.
You can always get into a project and then read a couple papers about the topic that you're working on and kind of get your way there and ask other people what they know about that topic. And it's a lot easier to kind of come back from that. Really, the meat and potatoes of research is being willing to keep with it, and stay on a project and not get bummed out when something doesn't go the way you expect, or being really curious about what you're working on. Because honestly, coursework is helpful, but it's more so an exercise in learning how to learn something, rather than you retain everything from that one particular class and it's never going anywhere. It's stuck in your head, and it's so you're gonna forget stuff, and that's the point. The point is that you can learn all this stuff and it's complex and you're familiar with it, but if you can learn stuff in the moment, then it's not such a big deal.
Yeah. Never be afraid to ask questions, because those questions will get you to where you need to go.
Yeah, exactly.
What tips do you guys have for people who are interested in applying for an REU, even if it's their first time?
I would say apply to as many as you can, as you can. REUs are really competitive, especially right now. I'm sure that they are so competitive because they rely on NSF funding. Point is, it's kind of like a numbers game. You apply to as many places as you are interested in, because it's also not you're trying to find a perfect marriage of exactly what you want to work on forever and ever and ever. It's just a place where you can have a research experience, get kind of acquainted with basic skills of that particular lab, get a sense of what conducting research is like, because that's really what those projects are for, and also to make connections with the people in those labs that you might go to, because they can be really great recommendation letters or contacts for when you're looking for a graduate position, maybe in the future if you're pursuing research in academia or whatever, really.
So where can people go to apply and find resources?
I forget the exact website, but it's the NSF REU. There's a dedicated website to it. It's a list of disciplines, and then
It's a site search.
Yeah. It's really a very, very large website. You just oh, I'm interested in geoscience REUs. You click on that, and there's pages and pages.
I know the Undergraduate Research Center is also a good point of contact for figuring out most things related to getting into undergraduate research.
I reached out to Laura Clippard, which I'm sure plenty of people are familiar with, if you know anything about the Honors College. And she was, a big help in starting to draft those first REU applications. There are plenty of resources, and also even if you're not associated with any organization, or if you're not associated with the Honors College, you can reach out to maybe faculty members that you have a particularly good relationship with, because they're very used to writing by students who are interested in doing research, or maybe they have some insight that they can provide.
I'd also recommend the Career Development Center for reviewing your CV.
Oh, yeah.
They were so helpful in getting my CV and providing feedback and helping me write it and my personal statement. It was awesome.
What was the most rewarding or exciting part of this experience?
I mean, for me, this is my honors thesis, so being able to not only get paid, but have the time and resources to do my honors project it's been amazing. Because I mean, to do it over the summer as an REU, and to even continue it throughout the semester, and even defend the work that I'm doing and present it at conferences, and it's amazing opportunity.
Mm-hmm. Yeah, I would say this seems kind of small in comparison, but my favorite part of each project that we do over the summer is when we finished a lot of the wet lab work. We're done growing cultures and passaging plates and stuff, and we get to take all of our data, put it into R, and just start feels
chipping away at it.
Yeah, start chipping away at it. It feels like a little spreadsheet game. Honestly, it's really satisfying. It's really nice. You get to reuse parts of code that you already wrote.
That's also a thing, though. I really didn't know that there was a lot of coding in research, and there is, and it's not that hard to learn it. And I was a little intimidated, but Ian is an amazing teacher. And yeah.
Well, I'm glad I could teach you. Dr. Arber, who teaches the biostatistics biometry course, she's a really great teacher. Does a really good fundamentals kind of course, and a higher up course for R coding, and she will talk about it the whole time. It is so important. I won't say it's mandatory, but it is incredibly helpful if you're interested in biology research at all to have some sort of basic understanding of R coding in R because one, it's comparatively to other, coding languages, it's not that hard, and it's really easy to like get resources, and really like it kind of just boils down to like recycling code from online, and also just like learning like, what am I trying to do? Okay, what is a way I know how to do it? And then kind of like creating this like horrible like piece together code script.
Right in stuff.
Right, yeah, signing scripts, that you kind of like whittle away, and you're like, oh, well, now I know a little bit better. I can do this a little bit more efficiently, and kind of just like, it's very iterative. I just might be a nerd about it, but it feels very satisfying to just sort of like fine tune little bits like that.
There are also resources. I know MTSU has a LinkedIn LinkedIn Learning. They offer R certification, so you can learn the basics in R, and that might be a good thing to consider when you're applying for an REU if you want to get like certification from MTSU and share that you have R coding experience.
It's an exceptional thing to pad your CV with if you like just do a basic training in R and get like a little bit familiar with it. Like, it really doesn't take a lot. You can put familiar with basics of R coding language, and then you put that in, and they're like, oh my god, we can work with that. It's never like a sense of like you have to be so good at it. Like, you really can just like literally know the basics and just like be willing to try and like piece together little bits of script, and like that's all you need, really.
Just sit at the laptop and you're just typing away, like, and then
that's what it feels like. But I'm really just copy and pasting the same couple lines of script that I'm reusing over and over and over again. It makes me feel so
and tweaking like a little bit.
Oh, exactly, yeah. And then it's like I've got like pages and pages of script. I'm like, wow, I'm so amazing. I'm so intelligent.
Tatyana, you mentioned earlier about conferences. You both presented at the Mycological Society of America over the summer, I believe. So how did it feel to share you all's work at a national level?
It was great. The Mycological Society is very focused on mushrooms, and we work a lot with bacteria, and so our audience was a little bit unfamiliar, and so it was really cool to be able to talk about that with them, because we're working with microfungi and not really like a mushroom. But it was super cool because we were recognized among like the undergraduates that presented there.
Yeah, we did well, which I feel like honestly partially is like owed to the fact that our poster was kind of a different topic and in like a slant that they're not used to at the conference. It's like a lot of posters about like the fruiting bodies of fungi and like mushrooms and like different like stuff you'd expect to see at a mycology conference. And then we're over here with like, oh yeah, our tubes of bacteria interacted with the metabolites of this fungi, and they're like, oh yeah, so like when they were growing with the fungi, and we're like, oh no, they didn't actually grow with the fungi. We grew the fungi for about 5 days, and then we sterilized it, and then stopped working fungi. But it is related because it's a bacterial fungal interaction. It's just an indirect one.
So if you had to summarize your summer in one word, what would it be and why?
Wow.
Wow, what was your word?
I was gonna say something else, but that was my word.
Nice.
It's a good word.
Yeah. Well, why?
I don't think can I explain?
Yeah.
Wow, because I don't know. There's so many different opportunities and experiences. Just being able to build on past experiences and create new ones. I think the summer was just wow.
Was your word why?
Yeah, we did ask that a lot.
Yeah, there was a lot of why moments. Yeah, honestly, that might be my official one, but I was trying to think of like a funny joke to go along with like dialysis and rotovapping. Yeah, no, maybe why, yeah, because like there was a lot of like, because we started with this like set idea of what we were going to do, and then we started, and then we kind of like hit a block where like, oh, actually we need to consider this other thing because the way we were thinking it was going to happen was not quite so simple. And then just like a lot of like back and like forth and like kind of like working our way through, we had to stop and think like, wait, like, why does this not work?
Why does this work?
What are we gonna do? Like, yeah, a lot of like stopping and thinking, which I feel like was a different experience from our first project, where I feel like we definitely had moments of like, oh, okay, we're not sure about this didn't work, or oh, we're just trying to keep it going. But it went a lot more like evenly. This one had a lot more kind of like back and forth and like sitting and thinking and like brainstorming, which is really fun. Like
something we needed, a bit of a challenge. Yeah.
I know, Ian, you mentioned you were going to graduate soon. So what's next for you guys academically or professionally?
Well, I hope to go to grad school. I've been looking at master's and PhD programs, like both in the United States and, in like Europe and other places, just kind of like seeing like what options are all out there. It's already like a pretty competitive kind of place to be at, looking for like positions, just because, there are a finite number of universities with open positions with funding for graduate students. But yeah, I'm honestly looking to kind of just like continue my experience, either like, potentially here or elsewhere, kind of like build on my skills already and try and take my time, make sure I feel like a really strong researcher. And then, I mean, as far as like long term, I'm interested in like, I am interested in academia. I'm also interested in like kind of working in conservation more directly. That would be an amazing opportunity to have.
So when you started your bachelor's at MTSU, did you think you would be considering grad schools in Europe?
No, definitely not. I don't know if I was even really considering graduate school. I'd always thought about it, but I just was a very unsure person going into undergrad. Like, I took a gap year because I was so unsure. But now this like experience has really kind of like taught me I really do enjoy research, and it's made me in some ways like a competitive applicant. I don't feel it, but I've heard it said before that like I'm a competitive applicant because I've had this experience in the lab these 2 summers and working during the semester, and I'll have like published work to present to wherever I'm applying to, and that's really exciting. So
my future directions are a little bit different. It's just because I hope to take what I know and apply it to human systems, because I feel like a lot of the times in medicine we immediately turn to like, oh, take this supplement or do this and you'll feel better. But I feel like where we kind of go wrong in that is that we miss so many opportunities to prevent it beforehand. Preventative medicine is like the biggest thing. And I think understanding the microbiome and applying that to how we think about disease and the health state of a person, I think is really important to evaluate.
See, when you said that you were going to be a little different, I was really hanging in there for you to be like, now I'm going to go be a coder for Rocket League. And
there's always time to pivot hard into a different direction. We could see you in like 10 years and you could be doing like visual art.
I'm going to apply R and make it like Rocket League R.
So now I have a lightning round. Pipette or microscope?
Pipette.
Coffee or energy drink?
Coffee.
Neither. Yeah.
Like, I live on water. I'm great.
Coffee just makes me really sick. I wish, though. I'm so tired all the time.
Favorite snacks? Well, do you have a favorite go-to drink then?
Oh, Arizona tea.
Okay. Yeah. Favorite snack?
I get into some like trail mix, like almonds, like cashews, like craisins. Like, I'll just be sitting and like eating it. Yeah.
Blueberries. I'm obsessed with blueberries.
Blueberries are great. Yeah.
Favorite class at MTSU?
Micro?
Oh, yeah. I don't know why I said that like I was surprised. I'm there. Right. It's very hard, but it's great. This is hard. Biometry. It was really fun.
What's biometry for those that don't know?
Biostatistics, yeah. It was the course with Dr. Arbor I was talking about, yeah.
Dream vacation destination anywhere in the world?
Switzerland.
I was literally going to I'm looking at universities in Switzerland right now, but yeah, my boyfriend went to Switzerland like a summer or 2 ago.
And he didn't take you?
Well,
Ooh.
It was a study abroad. I wish I could have gone, but I'm not in the geosciences department.
Get in the suitcase.
It's beautiful there. It's so beautiful.
It is in pictures for sure. Love to see it in real life.
Yeah.
So that wraps up today's episode of Honors Spotlight. A big thank you to Ian Wilson and Tatyana Martinez for sharing your incredible experiences and insights. If you're listening and thinking, wow, I want to do something like that, you can learn more about research opportunities, study abroad programs, and all things honors at honors.mtsu.edu. Make sure to subscribe to Honors Spotlight wherever you listen to podcasts so you don't miss future conversations with our amazing students. Thank you for tuning in, and we will see you next time.